During this webinar, I will give you an overview of the Cellvizio technology and introduce you to Cellvizio Dual Band, our instrument for preclinical research. I will then give you some application examples in drug delivery, cancer research, and neuroscience. But before I start, I'd like to take a couple of minutes to introduce you our company. The idea of founding Mauna Kea Technologies came from a multidisciplinary partnership between doctors and researchers in astrophysics. They aimed at developing revolutionary instruments using technology and expertise partially coming from astronomy. We are named after Mauna Kea, which is a volcano in Hawaii, but also one of the best sites in the world for astronomy observation. Our mission is to develop instruments that make it possible to visualize cells in vivo and promote the use of optical biopsy in order to improve patient diagnosis. So, what is Cellvizio? It is a unique, small and flexible microscope that can be used to perform imaging in vivo. The Cellvizio technology can be applied from the bench to the bedside. In fact, we have two business units, one focused on clinical applications. In this case, clinicians, they use Cellvizio as a decision-aiding tool for a wide range of indications in gastroenterology, pulmonology, and urology, for example. The Cellvizio Lab activities focus on applications in preclinical research. Scientists use Cellvizio for functional imaging or colocalization studies in the field of cancer research, neuroscience, infectious disease, etc. Thanks to these two business units, we can accompany you in any type of project, whether it is for basic research, translational research, during clinical trials, or even in the clinical practice. Now, the one-way formula from the bench to the bedside offers limited success. We are now more and more talking about cyclic translational medicine or reverse translational research, which gives more feedback from the practice to the bench. And thanks to this better communication, basic research has more inputs to improve personalized medicine. So Cellvizio will basically do what we call an optical biopsy of your sample. If you look at the standard tissue biopsy that is done in pathology labs, a cross-section of the tissue is stained and imaged using a standard microscope. The analysis is done ex vivo and the preparation of the sample takes some time. The Cellvizio optical biopsy will image the tissue directly in vivo and in real time by looking at the tissue from the point of contact. We are working with what we call confocal microprobes, which are made of thousands of fiber optics that are used to make the image of the tissue. So, for example, if you bring the microprobe on the top of the tissue, you will obtain a view of the horizontal section of the tissue. If you compare Cellvizio technology to other imaging techniques, you will see that we bridge a gap between whole-body imagers and benchtop laser microscopes. Whole-body imagers are compatible with in vivo analysis, but they have a low spatial resolution, while benchtop laser microscopes like two-photon microscopes can resolve submicron structures, but they can only be applied in vitro. With Cellvizio, you will be able to combine micron-scale resolution and in vivo measurements. Imaging techniques can be evaluated according to the following criteria: depth of penetration, real-time answer, size of the field of view, the sensitivity, if it's easy to use, and the resolution. Of course, the perfect imaging modality does not exist. If we want to evaluate Cellvizio imaging, then Cellvizio will bring you a high resolution, the ease of use, and the real-time answer. We also have a good sensitivity and depth of penetration. However, in our case, the field of view is a little bit limited. It ranges from 240 to 600 micron. If you look at PET-CT or Sept, they will provide you a maximum field of view and a very good depth of penetration and sensitivity. But in this case, the information is not given in real time and they offer a poor resolution. The use of such techniques will also require a highly trained operator, and also you will need to use radioactive material. The evaluation chart for whole body optical imagers will look like this one, and like this one for MRI. So what I want to explain is that only the combination of different modalities will bring you the perfect solution. We want to place Cellvizio as a technology complementary to optical in vivo imaging, to MRI, and to PET-CT. I will now quickly describe you the optics of the instrument. So the heart of the optical setup is made of two oscillating mirrors that you see moving here, and these mirrors are used to scan the incident laser light in the X and Y directions. We have a set of lenses that focus the light on the microprobe, and each microprobe is made of several thousands of optical fibers, from 10 to 30,000. The fluorescence emission of the sample is collected by the same fibers, and then it is sent to the detector. Cellvizio is a compact, turnkey, and very easy to use instrument. So what you see here is the Cellvizio Dual Band. Dual Band because it has a dual excitation and emission. We have two lasers working at 488 and 660 nanometers. We are talking about dual excitation and emission because the both channels are working simultaneously. So on the left you have the laser scanning unit, so this is where you have all the optical setup. Then we have the computer with the control software that allows you to control the laser scanning unit and to perform the real-time video recording of the fluorescence of your sample. And a key element of the instrument is of course the confocal microprobe that is attached to the laser scanning unit. We have different confocal microprobes of different diameters and different technical specifications. The smallest microprobe diameter is 300 micron. These confocal microprobes are flexible, so it means that you can insert them into a needle, a catheter, or an endoscope as it is done in the clinics. The coupling of the confocal microprobe to the laser scanning unit is very straightforward. You don't need to perform any optical alignment. It's a plug-and-play system that is universal to all the different types of microprobes that you may use. That's why we say that the instrument is very easy to use. Because we are working, we are probing a volume of several tens of microns, we can observe what we can call a volume effect. So in this example, you will see brighter structures which seem to be on the surface, while the other structures seem to be deeper. So this is the interface of the software. You have the possibility to, of course, control the instrument, so which laser you want to use, at which power, what are the acquisition parameters, and how you want to do recording. It is possible, for example, to perform time-lapse measurements. You also have the real-time video image display of the 488 channel, the 660 channel, and a merge of the two channels. In order to have a good image contrast, you can also play with the parameters from the lookup table. ICViewer is the name of the analysis software. We use it to perform kinetic analysis, which means to quantify the fluorescence signal over time on selected regions of interest. The data can be exported for reporting. We also have two optional modules to complement ICViewer. You have the possibility to perform mosaics. So it's like an image stitching tool in order to increase the field of view. The software will follow the position of the confocal microprobe over time, and it will rebuild the full image. We also have the vessel detection module, which is used to highlight and characterize vessels automatically. So this is an example of a mosaic performed on a mouse colon, and the cells were stained with acriflavine, which is specific for DNA. So that allows you to have a larger field of view. And this is another example obtained on the brain of a GFP mouse. So here you see neuron bodies and vessel branches. As for the vessel detection module, it can be applied on a specific frame, but also on mosaics. The user should only provide the diameter of interest, and the vessels are detected automatically, and the detection is based on the segmentation of the image. You get also statistics like total length or mean diameter. You can export these data into CSV files and generate some bar charts as well. And these features are useful when you are studying angiogenesis in cancer research, for example. Because the Cellvizio technology brings a new way of imaging your samples, it opens the door to a very wide range of applications. So we have applications in cancer research, in neuroscience, in reproduction, stem cell studies, vaccine development, etc., etc. So what I will do is that I will focus on some applications in drug delivery, in cancer research, and in neuroscience. In drug discovery, Cellvizio is a useful tool to study the biodistribution of a drug in vivo. In fact, thanks to the dual band technology, you can localize a tagged compound in vivo and look at its biological environment at the same time. In this example, we are tagging the vasculature from the intestine using phytidextrin, so in green, and a pharmaceutical compound was tagged in red with vivo tag 680. And on these videos, you can localize the drug, see where it goes, evaluate how it is absorbed very quickly. In this example, Cellvizio technology was used to monitor the local delivery of a drug in vivo. Ultrasound, they can enhance the membrane permeability, so they can facilitate the penetration of a drug into the cells. So the users have developed an in vitro setup. They have used Sonoview microbubbles to help generate the ultrasound, and they used cytoxgreen as a model drug. So cytoxgreen is a fluorophore that shows no fluorescence when it is in aqueous solution, but it will become highly fluorescent when it binds to nucleic acids. So basically when it will penetrate into the cells. So what happens when we apply the ultrasound under the setup? You will see that some signal appears. You see the fluorescence signal of the cytoxgreen. And these are the images taken at different time points, where you can see at the beginning no fluorescence, and towards the end a lot of fluorescence, because all the cytoxgreen has been uptaken by the cells. And if you also do a kinetics analysis, follow the evolution of the fluorescent signal over time, then you would be able to extract the kinetic parameters of cytoxgreen. Another application is the real-time evaluation of the blood-brain barrier. Being able to cross this blood-brain barrier is a key challenge for the treatment of most brain disorders. So in this work, the users worked on a rabbit, and they used fitsidextrin at different sizes to highlight the vasculature. So they induced the disruption of the blood-brain barrier using, again, ultrasound. So what happens, if you look at the video, what happens when you apply the ultrasound pulse? You will see a leakage in this area of the fitsidextrin. Here it starts, and now you see a leakage. So you get a confirmation that the blood-brain barrier is compromised. And then if we use the kinetics analysis, so here we have defined two regions of interest, one in red and one in green. So if you remember the video on the previous slide, this is on the red ROI that you will observe the breaking of the blood-brain barrier. So, and if you look on the graph at the evolution of the fluorescent signal, you can get the confirmation that you observe a local change on the top part of the image. So now we will look at some examples of applications in the field of cancer research. I think you know that angiogenesis is an important parameter in cancer research. In these applications, the users are working with AngioStamp, which is a molecular fluorophore to highlight alpha V beta 3 integrin, which is a molecular marker of endothelial wall and cells. So you can see on the video on the right the presence of AngioStamp in red on the control limb. But this integrin is also expressed, overexpressed in tumor neovessels. So it is an indicator of angiogenesis. You see two colors on the videos because phytidextrin was also used to highlight the vasculature of the tissue. So if you look on the left at the tumor, you will see first the appearance of the vessels. They look sinuous, and you have a lot of reds, showing that you have this integrin that is overexpressed. So it's an indication of angiogenesis. And the video in the middle shows that this molecular marker is cleared in the kidney as well. In this study from that was published in PNAS in 2011, the authors are working on pancreatic ductal adenocarcinoma, PDAC. PDAC is a fatal disease with very poor patient outcome because it is very difficult to diagnose PDAC at an early stage. There is an early stage form of PDAC that is called pancreatic intraepithelial neoplasia, but this stage, this neoplasia, cannot at this moment be diagnosed in vivo. So they have worked in a mouse model and checked whether it is possible to evaluate this early stage of PDAC in vivo. So they, because Cellvizio can be used in vivo, they have worked on using a longitudinal study, meaning that the same animal was analyzed at different time points. So they used the two fluorophores, fluorescein to look at the morphology of the pancreas and to evaluate also the extent of fibrosis as the disease evolves. And they also used another dye, ProSense 680, that highlights cathepsin's activity. And these molecules are indicators of tumor progression and invasion. So if you look at the videos, first looking at the morphology and the extent of fibrosis, you see, so in green, you see that at the beginning you have, let's say, normal structure of the pancreas, and as the disease evolves from the pancreatic intraepithelial neoplasia up to PDAC, you see that the tissue structure is completely destroyed and you have a lot of fibrosis. As for ProSense 680, you see that the tumor evolves rapidly and you have a lot of cell death. So this study is a real is very promising to develop a clinical tool to help in the early diagnosis of PDAC. So this slide is to show you what we can do for dynamic in vivo histology. We are calling it dynamic in vivo histology because we can look at the histology, at the cellular structure of your tissue in real time and in vivo. So the example here was performed on a mouse colon, and we are looking at the cells using acriflavine staining. So the dye is applied topically, and we use an endoscope in combination with our confocal microprobe. So the confocal microprobe is inserted into the endoscope. So if you look at the video on the right, you have the image of the endoscope. This is what you see through the endoscope, and you can also observe the tip of the confocal microprobe from the Cellvizio instrument. And at the same time, you get the image as a video, so it's dynamic, you get an image of the cellular structure. So you can evaluate and characterize, for example, normal mucosa, round crypts, and you have a regular size, and compare it to tubulous adenoma with larger crypts and an irregular size. So now we will look at some examples in the field of neuroscience. So this is an example where the users have been using Cellvizio to look at a peripheral nerve, and especially the regeneration of the nerve after crush injury. The standard technique is confocal fluorescence microscopy, but this technique can only be applied ex vivo, and it is performed on a fixed tissue, so you are losing a little bit the integrity of your sample. Also, you need to use multiple animals for longitudinal studies. For example, these images were obtained with a confocal fluorescence microscope, and they were obtained 4 days after the injury to see that the nerve is regenerated. Now, if you compare to Cellvizio technology, which can be applied in vivo on an anesthetized animal, you can use the same animal for the longitudinal study, and the same animal is its own control as well. So this is what was done. So the animal, you make a small incision into the animal and you insert the probe to evaluate the nerve. And at the beginning, so you have a control area where you see the fluorescence everywhere, and you have also the video obtained from the crushed nerve. So you will see on the video that there's a part where there is no fluorescence because the nerve was crushed. And after 4 days, you come back to the same area. You see on the control area, of course, there is still fluorescence, but also on the part where the nerve was crushed, you see a recovery of the fluorescence that confirms that the nerve is regenerated. So that was performed on the same animal. Regarding the evaluation of brain, two-photon microscopy is very limited and it cannot reach deep brain structures. You are limited to about 500 microns from the surface. But with Cellvizio, what you can do, you can access deep neurons in situ in a minimally invasive manner. What you can do is that you can insert the probe into deep brain. So this is an example that was used to perform calcium imaging in the hippocampus after bicuculin injection, so that induced epileptiform activity. So that this study was performed on a rat, and the users have used Oregon green as a marker for calcium activity. In parallel, the users have performed EEG monitoring. So the probe was inserted into the hippocampus of the animal, and if you look at the images on the top, you will see that some activity is observed. You see Oregon green that highlights, showing that after the injection of bicuculin, you have this epileptiform activity. And this is also confirmed if you look at the corresponding EEG. To be more specific and to look only at the calcium activity of certain neurons, it is possible to work with a genetically encoded calcium indicator, here GCAM3, and as shown in the movie individual neurons can be singled out based only on their calcium activity, and these can be monitored in real time. So this is the kinetics analysis, and you will see that each individual neuron lights up as the calcium activity is detected. In order to work with freely moving animals looking at their brain activity, we have developed a NeuroPack, and with this configuration, it is possible to insert the probe through a very small cranial window and to monitor the activity on the awakened animal. So you see the video at the bottom, the mouse is moving, and we can do the recording at the same time. So in this case, the length of the confocal microprobe is longer. It's 4 meters to allow the movement of the animal. In this case, so you see here the implant that is fixed on the head of the animal, and we have a holder to position the microprobe. So the microprobe stays in place. In this publication, the NeuroPack was used to monitor calcium activity using, again, GCAM3 in the ventral tegmental area, VTA, of a freely moving mouse. And they are working with fear conditioning, and they are looking at the plasticity of the calcium signals of the neurons. So this is an overview of the Cellvizio Lab product line. So we are focused on preclinical research. We have our instrument, the Dual Band, with dual excitation emission, 488 and 660 nanometers. We have a set of confocal microprobes that can be adapted to your application, Quantikits to calibrate the instrument, and the software to do your analysis. And so to summarize the content of this webinar, I'd like to remind you that Cellvizio is a complementary imaging device that will help you reach cellular resolution in vivo. Thanks to the confocal microprobes, access is no longer an issue. You can image from surface to deep tissues. The instrument is great for longitudinal studies. The same animal can be analyzed over time and for a long period to track the evolution of a disease or the effect of a drug. Our main fields of applications include functional imaging, deep brain, angiogenesis, GI disorders, stem cells, biodistribution, etc. So I would like to thank you very much for your participation to this webinar, and I will be very happy to answer your questions now.
Okay, so now we are back live for the questions. So do not hesitate to type your questions at the bottom right of your screen. We had some questions during the presentation. So first question, Aymeric, has it ever been tried to put the probe in a large blood vessel?
Yes, it has. So for the main application this has been done for is looking at atherosclerosis. So for that, if you want to look at the atherosclerotic plaque, it is better to go from within the blood vessel. You can also go from the outside, but you can go from the inside. So we have different types of probes, and some will go deeper to image, and so you can go from the outside of the blood vessel and look inside. Or you can go inside the blood vessel with small probes, but that depends on the animal. So obviously the bigger the animal, the bigger the blood vessel, therefore the easier it is for our probes to get inside the blood vessel. So typically you can't do it on a mouse, but you can do it on other animals. As soon as it's rabbits, for instance, it's pretty easy because our probes can go within catheters that are regularly used, or endoscopes or anything. So it's easy to do that.
Okay, thank you. Second question. So you talked about microprobes. So what type of microprobes do you have?
So we have three types of microprobes. One is surface probes. So those probes only have the optical fibers. They do not have any type of lenses at the tip. And so they will image directly from the point of contact of the tissue. So you will touch the tissue with your probe. That is the case for all the probes. And then you will image from 0 to 15 microns. So those three probes, not having any lens, can be very thin. So that's why we have the thinnest probes, 300 microns in diameter. The second type is the M probe. You can think of M as microscopy. And those have a better resolution. So it's down to 1.4 microns. So you can really see cells with very nice details. Mm-hmm. The last type of probes is the Z series. And those go deeper in the tissue and look at a bigger volume. So those are really the ones I was talking about to go, for instance, to look at inside a blood vessel from the outside. So three types: surface probes that are thin, probes that have a better resolution, and the last type, probes that look at a bigger volume, but at a bigger distance as well.
Okay. Next question. Is it possible to use Cellvizio for heart mouse analysis?
Well, that's never been done, just because it's pretty hard to access the heart. Yes. With one of our probes. So we have groups that work on the heart. Mm-hmm. It's bigger hearts. It's not mice hearts, and it's ex vivo. So they basically have a beating heart ex vivo and manage to put our probe in contact with the heart to get an image. Other than that, the point is really that you need to touch the tissue that you want to image to be able to get the image. So then the question is really how to access the heart and keep the animal alive to be able to do the imaging. So nobody has done that with our system yet, but really that is the technical point. Technically, our system would be able to get images from the heart, same as any other organ.
So talking about organs, is it possible to use Cellvizio to evaluate pancreatic tissue or tumor ex vivo? So,
sure. So ex vivo and even in vivo. Yeah. So to be able to access the organ you want to image, you can use the probe different ways. So you can either use the probe directly. So for instance, in the colon, you can just put the probe in there directly. Or you can use an endoscope, for instance. That is what is used with your clinical system all the time. You can use a needle, for instance, and put the probe through the needle. Or just make a small incision and put the probe directly in there to access the organ. So typically we showed you an example of a probe going to the pancreas. Mm-hmm. And so for that they used a small probe and a needle and went to the pancreas. So all the internal organs are accessible that way in a very minimally invasive way. Ex vivo you can obviously do it even more easily. The thing is, the probes are really there to be able to do that in vivo, but you can use them in ex vivo as well.
Okay, and I forgot one question. The preclinical probes are dedicated only for animals, or can they also be used on humans?
Yeah, so those are very separate things. So it's the same technology, but there are two different systems. Mm-hmm. The clinical system is really made to be only used on humans, and the probes that go with that system, the clinical probes, are dedicated to human use as well. They have a limited number of uses, but they're dedicated for that. For the preclinical system, the preclinical probes that go with it are only to be used on animals in a limited number of times, but only on animals.
Okay. And so what is the sampling rate For example, for calcium imaging, it is the limiting factor for, is it a limiting factor for calcium reporter?
So that's a good question.
Yeah.
So indeed for calcium imaging, so by default our frame rate with the system is 9 images per second, and we can go up to 40. So depending on what you want to do, as far as calcium imaging goes, you will be able to do it easily or not. So you will be able to go up to 40 images per second, as I said. So depending on the reporter you use, it will be the limiting factor, or the system will be. So yeah, it really depends on the type of neuron you're looking at, where in the brain, and so on. So it's really dependent on that.
Okay, thank you. Another question regarding the vessel detection software. Someone wants to know if you can accurately detect the vessels if you have some movement from the breathing of the animal.
So there's two parts in that question, I feel. So the first is you do have that motion from the breathing of the animal, and wherever you are, that can be a problem. So it really depends on the application to be able to keep the probe on the organ you are imaging. And for that, you can use micromanipulators. Where you're in the brain, you will use a stereotaxic frame, or you will hold the animal when it's anesthetized. There are multiple ways. So that's one thing. Then the vessel detection part, that is only on one image at a time. So the vessel detection module does not follow the evolution of blood vessels with respect to time on the video you are recording. So what it does is it detects the blood vessels on one image, and then if you want to compare with another image, you have to use it again on that specific image to be able to detect those vessels and then compare. But since it won't follow from one image to the other, the breathing motion won't be a problem for that module specifically.
Okay. Another question. So I have lots of questions. What is the benefit of Cellvizio compared to a two-photon microscope?
So there are two main things compared to a two-photon microscope. The first one is we really do in vivo imaging. So a two-photon microscope can do in vivo imaging as well, but it is limited to basically 500 to 800 microns as a depth of imaging. And what we can do with our probe is really go anywhere inside the body and image at that point. So typically inside the brain, we can really go deep inside the brain, really anywhere, including the VTA, for instance. And that's thanks to the probe that can penetrate the brain. But that's the case for any organ in the body, so you're not limited to that surface. The last thing is also that we can do longitudinal studies because it is really minimally invasive because we have small probes that go inside the body. So you don't need a full window or a big chamber the same way you do with the two-photon microscope.
Okay. I have lots of questions regarding the use of dual bands in humans.
Okay.
Can you comment?
So as of now, the dual band system is only available for animals and not for human use. So for now, only the one color system, 48 nanometers, is really available on humans. The two color system is really only for animals for now.
Yeah. A question: is it possible to detect blood flow and velocity using Cellvizio?
So that goes back to our frame rates,
Yes.
and really how you label what you want to image. So in the case of blood flow and velocity, as far as I know, blood flow will be faster than the 40 images per second that we offer. So with that, you won't really be able to measure a velocity of blood flow, especially in small animals. You will be able to follow some cells For instance, some leukocytes go really slowly when they follow the wall. So you might be able to follow them, but not the full blood flow. So you will see the blood flow, but measuring the velocity, we don't have as of now enough of a sampling frequency.
Okay, thank you. So I think now time is over for questions. There are more questions coming. We will answer to your questions by email. If you want to contact us, you can visit our website, cellviziolab.com. You will find some information about the product and also applications and conferences. For example, we will be exhibiting at the Society for Neuroscience next month in Chicago. So if you are attending this conference, do not hesitate to meet us at our booth. If you had some issues or you would like the recording of this webinar, you will receive an email, probably before the end of this week, with a link to watch the recording. Thank you again for joining us today. To make this webinar a success, we had a lot of questions, a lot of interaction, and we hope to see you soon. Before you leave, I would just ask you to take just 1 minute to fill in a survey that I have prepared. It will help us improve our webinars and find new topics for you. So don't hesitate to write which topic you would like to be covered in the future. So thank you again. Thank you, Aymeric. And I hope to see you soon. Bye-bye.
Bye.